Hello, everyone. My name is student Dr. Pham, and welcome to my presentation on the effects of PXR activation on cancer cell viability. And this was a project done under the mentorship of Dr. Bradley Creamer. So continuing on with the PXR train from previous two presentations, hopefully by now everyone has a pretty good understanding of what PXR is and its biological roles. Now I want to bring everyone's focus onto PXR implications in carcinogenesis and breast cancer. So studies have shown that PXR have been linked to the carcinogenesis of several cancer types, like breast cancer in MCF7, T47D mentioned in Jack Abbey, Brandon Yasmin's presentation. It's also been detected in liver, colorectal, ovarian, prostate cancer, etc. And on the right side, you can see that these are all the biological roles that PXR can be involved in carcinogenesis. And one thing to keep in mind is that PXR effects are highly cell line specific. So we cannot use data set from one cell line to interpret data set from other cell lines. But with that being said, PXR has opened up a very exciting new avenue for anti-tumor therapy, and that is what we're trying to do in this lab. So here, I want to introduce everyone to our hypothesis for the experiment. We hypothesized that the activation of PXR by an agonist, either rifampicin or SR, can influence cancer cell viability significantly when treated with chemotherapy. And before I go on to show the viability experiment data, I want to quickly mention our QT real-time PCR on the mRNA expression of PXR in different cell lines. So we use LS180 as our reference group because it has decent endogenous PXR expression. Represented in the green circle here is MCF7 PXR expression. It has slightly higher mRNA of PXR compared to LS180, but MBA, MB231, and T47D have slightly lower mRNA expression of PXR compared to LS180. And this influences our experimental design, which I'll show in future slides. So for our viability experiment, we use an assay called Alamar Blue. This comprises of a blue substance called risazorin, and this can be reduced by NADH produced by active metabolism of cells to turn into a pink substance called risazorin. And both of these can be picked up by measurements of absorbance at 570 nanometer and 600 nanometer using spectrophotometer. And when we have the data, we can plug it into a specific formula to allow us to quantify the amount of live or dead cells in the treated groups compared to no treatment. And so we first did this experiment in LS180. And again, LS180 has pretty good endogenous PXR expression. So that's why we picked it first, and then we didn't have to introduce any exogenous PXR into these cells. We used 96 well plates to culture these cells and treat them. And we have three different experimental groups, one with chemo treated only, one with agonist plus chemo, and one with vehicle, which is DMSO plus chemo. And then we use 8 different concentrations of chemo drugs. So this timeline here shows how we did it in 1 week. We first started on Wednesday with seeding the cells at 5,000 cells per well density, and we made a total of 4 plates because we used 2 kind of chemo, cisplatin and doxorubicin, and we used 2 agonists, rifampicin or SR. So we let these cells incubate for 24 hours and then treat them with PXR agonist, and another 24 hours before we treat them with chemo, plus or minus agonist or vehicle, depending on their experimental group. And then all these cells were incubated for 72 hours over the weekend before we incubated them for 4 hours with Alamar Blue and then measured the absorbance. And that allowed us to identify the IC50 for all the treatment groups. So IC50 or inhibitory concentration, where 50% amount of cells in the treated group were killed compared to no treatment groups, is what we're interested here, because when we compare the IC50 for the group with the addition of rifampicin and SR, we saw that the IC50 were lower in these groups compared to others for doxorubicin, which means that lower amount of chemotherapy were required to kill 50% amount of cells in the treated compared to no treatment. And the same pattern was seen here in the cisplatin group as well. That was very interesting. So we decided to do that on repeat experiment on T47D breast cancer cells. as per our mRNA data, T47D has slightly lower amount of mRNA for PXR, so we decided to introduce exogenous PXR, GFP, or GFP only into these cells. Most of the experimental design were kept the same as the previous experiment, but we switched to a 24-well plate in order to avoid overcrowding of cells because of one extra day for the transfection, as we can see here. We also reduced the amount of agonist and chemo used, so only narrowed down to doxorubicin and SR, but everything else kept the same. And here are our data. So on the left are the IC50 curves for all of the groups, with GFP PXR group shown in green, and then GFP only shown in orange. And then on the right side, I isolated the IC50 curves for doxo plus SR in the GFP, PXR, and the GFP groups. And as everyone can see, this gap in between these two curves. And in the red box shown up here are the IC50 values for these two groups. So we can see that the addition of PXR agonist lowers the IC50 for doxorubicin in T47D cells. In the future, we plan to explore other methods to quantify cell cytotoxicity, for example, using MTT assay to validate our data, as well as repeating the experiment in other breast cancer cell lines to further understand the effect of PXR activation in them. And with that, I want to thank KCU Office of Research for facilitating SSRF. Special thanks to Dr. Staudinger, Dan, Abby, and Ambrose for contribution to data, and other MKRC Lab members, Brandon, Jack, Yasmin, Amalika, and Philip for their excellent support. And here are our wonderful people. Thank you very much for your listening. I'll be happy to.
It's great.